This is 6.3.
This is
6.3. Take
6.3. And you don't have to do 6.4. We're not doing 6.4. Here's AP Bio right here. Guy. All right. You need to understand this graph. This graph has energy on the left, free energy. You can just think of it as energy. And the progress of a chemical reaction on the bottom. And there's a bunch of lines. There's a couple of lines here. Well, you should be familiar with this graph from chemistry. Did you ever see a graph like this in chemistry class? Those of you who've taken chemistry?
I don't think so.
Well, you should have. This shows the energy, the activation energy. Did you ever talk about activation energy in chemistry?
Yes.
The activation energy is when you have any kind of chemical reaction, it usually takes some energy to get started. Are you filming now? Good for you. And are you ever focused on this up here? Okay, so they could be.
Thank you.
When you carry out a chemical reaction, and a common chemical reaction is burning something, right? Y'all know about this, right? You go during school and you go out to the car and smoke cigarettes, right? So what we're talking about there is you set something on fire, and then once you set it on fire, the thing kind of burns on its own, doesn't it? And all of that can be, you can think of it as this graph. So the reactants, that's what you start with. That's what you start with, and then if you're burning a piece of paper, it's the paper is the reactant. And also, you know what else is involved in the burning reaction? Oxygen. Very good. You got the paper and oxygen. That's the reactants. Now, just by themselves, they're not gonna react very easily. But you can give them what's called an activation energy. And you can give them extra energy. So here's the energy that the reactants start with, and then you give them some extra energy. And once they get that extra energy given to them, that makes them react together. That makes the oxygen combine with the molecules of the paper. And that's an exothermic reaction because it forms some final products. And here's the products. The products are at a lower energy than the reactants were. So if the final products are lower in energy than the reactants you start with, energy must have been given off in the reaction. Kind of a little chemistry review. Does any of this sound familiar? So where does that energy go? Well, that's the fire that's coming out of the paper that's now on fire. That's the energy coming out from the reaction. But you got to give it a little activation energy to get it going, and that's the little when you light the paper. After you light it, it'll go on its own because the energy that's given off is used. Some of the energy that's given off is used to start more reactants is used as activation energy to get more reactants to become products.
So
and the thing just carries on by itself.
So exergonic means that the final products have a lower energy than the reactant.
That's so well said. I love that. Exergonic means the final products have less energy than the reaction. And so when it's going from this state to this state, when it's going from paper and oxygen, you know what the products of combustion are? CO2 is one of them, and H2O is the other. So the paper and the oxygen become carbon dioxide and water. That's what happens when you burn paper. And they just float away in the air. Isn't that interesting? Now, here's the thing about this chapter in biology. We can get this reaction to go a lot faster. We can lower this activation energy to a much lower state and get the reaction to go a lot easier and a lot quicker if we have what's called an enzyme present.
Speeds up the reaction.
Speeds up the reaction. It's a we say it's a biological catalyst. You may have heard that word. A catalyst is something that speeds the reaction up. So here's an enzyme. An enzyme is a protein. Remember the protein that I had? A bunch of amino acids together that fold up into a certain shape. And that shape is just the right shape. In this case, it would be the right shape to accept molecules from the paper and to accept oxygen molecules, and they fit in the enzyme in a certain way, and that causes them to connect and form carbon dioxide and water. And it can happen at much lower energies. You don't need a flaming lighter to get it to go. All you need is this enzyme, and that can cause the reaction to happen without much activation. Let's see some video footage.
In every chemical reaction, the reactants pass through a transition state that has a greater energy than that of the reactants or products alone. The difference in energy between the reactants and the transition state is called the activation energy. If the activation energy is available, the reaction can proceed, forming products. An enzyme or other catalyst reduces the energy required. It provides an alternative transition state of lower energy, called the enzyme-substrate complex, and thus speeds the reaction. Notice that the catalyst decreases the activation energy, but does not alter delta G, the free energy change in the reaction.
Yeah. So we call the difference between there, where the reactants were, and the products were, we call that delta G. Delta G is the change in free energy. G means free energy. So there's a change in free energy. It goes from energy reactants to energy of products here. This is an exothermic reaction. Can you draw me what the reaction would look like if it's endothermic? Can you draw me a little line like this if it were an endothermic reaction? What it might look like? You and your group, in 10 points, if you can draw it for me. What do you think it would look like if this was an endothermic reaction? All you got to do is draw a line. This is an exothermic. See if you can use your brain and your buddies. Think about what an endothermic reaction would look like. Remember, an endothermic reaction requires energy. It doesn't give off energy.
I need to put up more paper.
Like,
No.
like what? Here.
Do you have any idea?
It means that the energy of the
Being like, well, it's probably going to be like...
Probably going to finish higher.
Anyone?
I don't know if it would. What would be like?
Yeah, first session.
Yes. Scissors.
Scribble the other side. Like that?
You're welcome. Anyone else?
I think something more.
Oh, green?
You can do it on here.
Oh, green's got it.
I was thinking something more like...
Green, 10 points.
Oh, let me write it up.
Blue.
No, then.
I'm not going to give it to blue. Oh,
No.
come on now.
Like this?
No, I'm not going to give it to blue.
Other side.
Yeah.
Okay, I'll give it away. Anyone else?
We're coming. We're coming.
I'm not counting it wrong on blue. Going.
Here, wait. We need 1 second to do this.
1.
Wait.
Second. I used to tell that to my
Okay,
sister.
here.
She'd say 1 second.
Wait, sorry.
And then she'd start crying.
So that's an exogonic,
This is an exergonic.
but it's also
Yes, Peach is correct.
It's also a endothermic.
No, it's exothermic. It's exothermic and exergonic. Those terms are similar. What this would look like, do I have a picture here? No. I don't know.
When molecules collide, there must be...
Oh, wait. that was it.
The minimum collision energy for the reaction to
See,
occur.
that's what it would look like.
Lacking this energy, the molecules remain intact and no change results from the collision. The colliding molecules must have a total connection.
So you can see if the products are at a higher energy than the reactants, then it must have taken in energy to get that reaction to go. In this kind of reaction, an endothermic reaction wouldn't feel, you wouldn't have heat coming off of it. It would feel cold. It's taking in heat from the environment to go. And the reaction vessel would feel cold if the reaction was going on. Yes?
So why does it go down after it reaches its peak in endothermic?
So there's a activation energy that has to be given to get these two things to react. And that's at a higher energy. But then that'll come down to this level. But that level is above where it started here. So overall, there is an input of energy. Here, the level is well below where it started. So overall, there's an output of energy.
So there's activation energy for both exit products?
Both of them need activation energy. That's
Okay.
correct. Yeah.
Okay, so when the final products have a higher energy than the reactant, that means it's endergonic?
Endergonic or endothermic.
So
Yeah.
they mean the same thing.
Basically, yeah, for our purposes. There's some differences, but it's a chemistry thing. We won't worry about it. Okay? So, here's an enzyme, and you need to know enzymes are shape dependent. The shape of an enzyme is what's important. You with me over here, Cam? Up here, this way. There you go. Enzymes are shape dependent. So here's the enzyme, and again, it's almost always a protein in a cell that has a certain shape. Imagine this protein had that shape. And there's the substrate, that's what we start with. That's like the reactants in a chemical equation.
So the substrate are reactant.
Yeah, the reactants. What starts the reaction. And here they fit, the substrate fits together with the enzyme. See, they fit together? We call this the lock and key mechanism. Because it's like a key fits into a lock. The key has to have the right shape to go in there. Josh, you with me here? Yes, question.
Okay, so when you're doing like the graph again,
Yes.
like when it's endothermic, would the enzyme there,
Uh-huh.
would it be higher still?
Well, this hump would be lower if an enzyme was there.
Okay.
Less activation energy.
Okay.
Okay? So enzymes just lower the activation energy hump.
Okay.
Make sense? So here we go. Here's the substrate. It fits into the active site of the enzyme. We call it the active site. It's the location where the substrate fits. And it just clicks onto there. And now we have what's called the enzyme-substrate complex. It's enzyme and substrate together. They love each other. They're hugging real tight. Then the substrate, in this case, is broken into two pieces, and there you have your products of the reaction. That's chemical reaction. We started with that, we ended with that, and we had to go through an enzyme to do it. This substrate could come apart into those products without an enzyme, but you would need a much higher activation energy. You would need just something that can tear those things apart. You need to get them shaking real fast, maybe raise the temperature up, and then they'd come apart on their own. But if you want to do it at a lower temperature, you put an enzyme in there that has just the right shape that when that substrate lands in that enzyme, that little bump there just kind of wedges the two of them apart, maybe. And the pieces come off easily with an enzyme present, but they don't come apart easily without the enzyme present. Does that make sense?
So degradation is when the substrate is broken down.
to smaller
Product.
Yeah, see? So it comes apart into two pieces, and here's a synthesis down here. Here's two pieces being put together by an enzyme. Some enzymes put things together, some enzymes take things apart. Which would you be if you were an enzyme? A putter together or a taker aparter? Anyone? Drew, what would you be?
Put it together.
You're a putter together? I'd take you for a taker aparter. I'm a taker aparter myself. Want to see video footage? Oh, there's no sound.
On the enzyme, called the active site, has a shape that fits with specific substrate molecules. An enzyme works by binding to one or more specific molecules called reactants or substrates. Binding occurs at the active site. The enzyme and substrates form an enzyme-substrate complex. The interactions between the substrates and the enzyme stresses or weakens some of the chemical bonds in the substrates. These stresses encourage a link between the two substrates, leading to the formation of a different molecule. As a result of the chemical interactions within the active site, a new product is formed. The product is released from the active site. The enzyme assumes its original shape and is free to work again. Although this reaction has specifically illustrated the formation of a single product from two substrate molecules, other enzymes catalyze the formation of two products from a single substrate.
Okay, so just real fast. Here's the enzyme. Here's the parts coming in. They click into place, and the enzyme helps cause them to come together, and then they leave. Huh? Isn't that exciting?
Mm-hmm.
Okay, so here's some questions for your group. You ready?
Mm-hmm.
What do we call those? That's question one. That's A. And on the same sheet, what do we call this? That's 2. This whole
That's
purple
B.
thing, the entire purple thing, that's B. And C is what do we call this area where these things come into this purple thing? 5 points each. So what are those? What is that? And what is this area called?
Does that look right?
5 points.
I'll take it.
Each right on all 3. Green right on all 3. White right on all 3. Blue right on 2. Get that right. I'm a poet and I don't realize it. Purple right on all 3. Okay, so these are called substrates. This is the enzyme. And this area here is the active site. Just see if you're paying attention there. Now, notice the active site kind of changes shape a little bit. Watch it change shape. Whoop. See it change shape a little bit? We have a name for that. It's called the induced fit model. Induced fit. Induced fit says that the enzyme changes shape a little bit when the substrate attaches to it. Induced fit. Now, enzymes are always specific to where they where they work the best. Are you with me, Nick? Pepsin is an enzyme that is very active at pH 2. You know where you find pepsin? Cam, hang with me, man. Anyone know where you find pepsin?
Pepsi.
Pepsin is in your stomach. It's at very low pHs. That's a digestive enzyme that works in your stomach. And you can see that it's very active at pH 2. We say that's its optimal pH. Trypsin is found in your intestine, where it's a lot more basic. See, that's very active at pH around pH 8, where it's more basic. Every enzyme has its own specific pH where it works the best. We're going to do an experiment starting tomorrow and continuing Thursday, where we figure out what pH an enzyme works best at. You're going to have to figure it out. I'm not going to tell you what pH it is.
Why is 11 bold?
I have no idea.
What did you call it? What did you call it? Optimal pH.
An optimal. Optimal means the best. Works the best at. Here's a look at temperature. All enzymes have an optimal temperature they work best at. Most animals, the temperature is about 35 Celsius. That's about our body temperature, 98 degrees Fahrenheit. Most animals' enzymes work best about that temperature. Here's why. If it gets colder than that,
it needs more energy.
Well, yeah, the molecules aren't moving very fast. And they don't move very fast and come into the active site very fast. They're not broken down very fast. If you're warmer than that, you know what happens to the protein? What happens when
Breaks
you get real
apart.
hot in your protein?
You get radioactive gas.
There's a word. 10 points if you can tell me what the word is for what's going to happen to this protein when it gets too hot. You should know that word from a previous. If it gets too hot, the enzyme kind of shakes like this and comes apart. We have a name for that.
Sure.
I contribute.
White. Correct. Peach. Correct.
Good job. I didn't know that.
Green. Correct.
You got it.
Anyone else?
Y'all got this.
It's called denaturing. The protein comes apart if it gets too hot. So look at this. Above about 35 degrees, proteins denature and the enzymes quit working. That's why you can't live. You can't dive in boiling water. You'll die because all your proteins will denature. And your cells can't survive if they don't have enzymes doing their jobs. That happens very quickly when you get to high temperatures. So at high temperatures, proteins denature. At low temperatures, molecules don't move very fast. All enzymes have an optimal temperature at which they work best. And it's usually just below when proteins start to denature. Look at the polar bear. Look at the cat. I don't know why these pictures are on here. Here it shows a protein working. Sometimes proteins require what's called a cofactor. A cofactor is a substance that kind of fits into the active site and makes the enzyme work better. It often makes it the right shape. Have you ever read the side of a cereal box?
Yeah.
And you see all those things like calcium and niacin and iron and all these things you need, and you never knew why they're necessary. A lot of those little elements, like you wouldn't think you need, iron might act as a cofactor for some of these enzymes.
So it just helps the enzyme.
Helps the enzymes work. So you need those. I'm just drawing blanks of what's on the sides of cereal boxes. I used to read those every morning. Give me some things that are on the sides of cereal boxes. Anyone?
Magnesium?
What's that? Magnesium, yeah. Okay.
Really funny. That
Magnesium might be a cofactor.
really fun.
Sometimes the things you read are, the vitamins are also called coenzymes. Here's an enzyme, and here comes a vitamin, which is a coenzyme, and the two have to be together. There's another one. Oh no, that's the substrate. So the vitamin, which in this case is thiamine pyrophosphate, that's a vitamin that you need in the diet. There's an enzyme. The enzyme cannot be active without that vitamin. This is why you need to eat your vitamins. And now here's a substrate, fits onto it, and then comes apart, and there's your products. So this enzyme wouldn't work without the vitamin. Here comes another one. This is what we call a coenzyme. Is a molecule that helps an enzyme work. Cofactors and coenzymes are both molecules that help enzymes work. So here comes a coenzyme, and look, it gives the enzyme the right shape so it can do the reaction. And the enzyme can just do one reaction after another. Oh, so here's some of the things. Thiamine, riboflavin, niacin, vitamin B6. These are all coenzymes that help enzymes work.
So what's the difference between cofactors and coenzymes?
Good question. Cofactors are inorganic. They don't contain carbon. Coenzymes are organic. They do contain carbon.
So what does that really mean?
An enzyme is a biological, is a big molecule. A coenzyme is a larger molecule that has carbon base.
Okay.
Carbon can form big molecules. Cofactors are inorganic. They're always small, a couple of atoms. So I got all kinds of videos here showing enzymes.
Catalysts are substances.
That's a bad one.
An
Here's a good
enzyme
one. Watch
attracts the reacting molecules or
Just the aids
substrates
and like
to its
the
active
enzyme
site,
process or whatever.
then releases the end products. Its
Yeah, it's
effect
good.
Watch
is
it. You gotta watch the
astounding.
video.
Each enzyme can assemble hundreds of thousands of new molecules every second.
There are enzymes that carry out 1 million reactions a second. How fast these molecules are moving. They just come in there and phew. See, we have the reactants coming in. They fit into the active site. Enzyme changes shape a little, and there's your product coming out. That's how chemical reactions happen in cells.
Substrate molecules
Here comes some coming in. They fit together, and then they come out.
Enzymes.
Here's an enzyme. Here comes some molecules coming in, the substrates. They fit on it just right. It's got to have the right shape. The enzyme bends a little bit, and now they're together. Are y'all getting the point here?
Yeah.
That was what we just did.
According to the induced fit model,
Substrates coming in to the active site. They fit in there, push together, induced fit, and these never come out. What a lame video that was. Here comes sugar and water, sucrose. You ever heard of sucrose?
Mm-hmm.
Can you tell that's a sucrose molecule?
Yep.
Right there? Two kind of hexagon
No,
and a
I
pentagon,
really
and there's
can't.
water. And watch, they fit into this enzyme. Let me go do that again. Here we go. Fit into the enzyme, sucrose and water, and then they come out, and now you have glucose and fructose. Sucrose and water form glucose and fructose. That's an enzyme in your digestive system that breaks down sugar.
So that was a hydrolysis reaction?
Oh, Nick, that's so good. That was a hydrolysis reaction. Want to see it again?
Do it, Nick.
Here comes some water. Little hydrolysis. There you go. Did you like that reaction?
What?
Oh, here's my favorite enzyme. It's bovine pancreatic trypsin inhibitor.
I was going to say that too,
Pepsin
but she
digests
didn't.
proteins by hydrolyzing peptide bonds.
There's some people that are in trouble. I don't know what's going on there. It's just craziness. Okay. What this last thing shows is that these enzymes work in groups. It's not just one enzyme usually doing a reaction. The enzymes usually work together. Drew, are you with me? Hang with me, sir. Because this is fantastic. So here we have 4 enzymes. And they're all going to work together. And I'll just let this run
Organisms
through.
contain.
And I'll take the audio out. So here's 4 enzymes in a row. And we're going to see lots of examples of how enzymes work in groups. But here comes a substrate. A substrate is going to come into an enzyme. And get ready, because it's going to come. Here it is. That's the substrate. And it's going to fit into the active site on enzyme 1. And then it's going to come out. Then it's going to go to the active site on enzyme 2. And that's going to change it even more. See, often you need to make several changes to items to get them to make some final product. That's product 2. That's product 3. There it is. And finally, number 4. And there we have our final product. So we had to go through 4 enzymes to get this made. And where you see this sort of thing is in the Golgi apparatus. Remember the Golgi was stacks and stacks of membranous sacs? Mm-hmm. I will not eat green eggs and ham. I will not eat them. Tom, I am. Because the first sac has one enzyme, and the second sac has a second enzyme, and the third sac has a third enzyme, and the fourth sac has a fourth enzyme. And after you finish, go through all those stacks, you have a final product.
So the Golgi apparatus is like the enzyme.
But
Enzymes are inside the Golgi.
But isn't that just one reactant?
Yeah, but it could be more than one.
Okay.
This is just showing you the simplest way it happens, but it could be more than one reactants come in to form final product.
So is that an induced fit enzyme?
Induced fit, yeah, is when the enzyme changes shape a little bit. I don't think it's showing that. It's not showing it change shape, but it actually does.
What's the
Putting in
what
work.
is it when the substrate changes shape?
Well, it's just changing into something different.
Working
Maybe
hard
there's
or
some
hardly
water
working?
and stuff being added that make it change. It's just trying to simplify it for you. Now here
Many of the enzymes.
Here we have the substrates coming in, and it's going to form a final product just like before.
Isn't this the same video?
Here's the final thing, pay attention. Once our products form, there has to be a way of turning this off. I mean, how do the enzymes know when to stop making their products? Once we have enough product, we need to shut the pathway off. So there's a mechanism for that too. These products will be just floating around everywhere, and some of the product will randomly float around because molecules are always moving. And watch this product just randomly floats around, and eventually it'll hit a site on the first enzyme in the chain, like it just did there. Let me back it up a little bit. This is one of the products are forming over here, and one of the products comes over here and hits a special site right here. That site has a name. It's called the Allosteric Site. The allosteric site is a site on the enzyme away from where the active site is. Here's the active site up here. But the allosteric site, when the product reacts with it, so the final product comes in here, sticks to the allosteric site, and that's going to change the shape of the active site. Watch the active site change. Here we go. It binds. Watch the active site change. Whoop. Did you see it close up? And it can no longer accept substrate. So we stop forming products. And that happens automatically. Once we have enough product, some of the product will just happen to bind to the allosteric site on the first enzyme in the chain, and you won't make any more product. You've shut it off because you've made enough. Cells will automatically shut down their processes when they make enough product. And only when this product is used up, when the cell has used up all of its product, it'll use that one up too, and that one will fall off, and when that one falls off, the enzyme goes back to the right shape, and you can make more product. Isn't that a cool system? Mm-hmm. It doesn't have to think. When it needs more product, it automatically turns on, and when it's made enough product, it automatically shuts off. Enzymes are awesome. And what we're going to do is a lab tomorrow where we take an enzyme from a turnip root, and it breaks down hydrogen peroxide. And we're going to pour some hydrogen peroxide in a test tube, and we're going to put some enzyme in a test tube, and it's going to break up the hydrogen peroxide, and it's going to form water and oxygen, and we're going to put an indicator in there, and we're going to see a color change when that happens. And you're going to see what pH that enzyme works best at. By some pHs, it'll show hardly any color change. Some pHs will show a lot of color change. The ones that show a lot of color change have the enzyme working fast. Am I talking too fast for you?
No.
Sometimes I talk fast.
So
I'm
this
excited
test-
about enzymes. They're so cool.
Is this test 4 through 6?
This test is 4 through 6. It's going to be Friday.
Wait, is this test on Friday?
It's on Friday.
Don't we have short classes on Friday?
Yeah, we'll have a short test.
We need a short test.
But usually they're like 35-minute classes.
35?
35
Uh-oh.
minutes. Yeah, because there's a spirit.
Are you
What's it
serious?
called? Yeah.
Maybe we'll just have the multiple choice and do the essay later.
They're either 40
Yes.
minutes.
Probably do that. How about that?
So it's just multiple
On Friday we'll do the multiple choice, and I'll do the essay on Monday.
What is excluding 6.4?
Yeah, you don't have to do 6.4. We're doing 6.4 later. So, okay? All right, peace out.